Welcome back to our new video, that is MCQs on biochemistry, and this is the part 5 of this series of videos on biochemistry. So the first question of this video is, which of the following diseases are associated with genetic or nutritional deficiencies that alter protein maturation process? And the options are option A, Kuru, option B, Scurvy, option C, Creutzfeldt-Jakob disease, or option D, all of the above. So the right answer is option D, that is all of the above diseases are associated with genetic or nutritional deficiencies that alter protein maturation process. Okay, so let's move on to the next question, that is question number 82. So which of these sets of amino acids are not capable of forming hydrogen bonds through their side chains? And the options are valine, isoleucine, phenylalanine, or option B, tryptophan, tyrosine, histidine, option C, serine, threonine, asparagine, or option D, arginine, lysine, aspartic acid. So the right answer about this is option A, that is valine, isoleucine, phenylalanine. So these three amino acids are not capable of forming hydrogen bonds through their side chains. So they don't have side chains containing hydrogen containing OH bond, OH molecule or compound, so they cannot form hydrogen bonds. Okay, so let's move on to the next question, that is question number 83. So hydrogen bonds in antiparallel beta sheet occurs in more numbers than van der Waals interaction, or option B, are not present at phenylalanine residues, or option C, occurs roughly perpendicular to the polypeptide chain direction, or are about 5 angstrom in length. So the right answer about the hydrogen bonds in antiparallel beta sheet is option C, that is it occurs roughly perpendicular to the polypeptide chain direction. So this is the right thing about the antiparallel beta sheet. Okay, so next question देखेंगे. Next question is the hydrophobic moment is 0 for amphiphilic secondary structure, or negative for amphiphilic secondary structure, or option C, positive for amphiphilic secondary structure, or option D, intermediate for amphiphilic secondary structure. And the right answer is option C, that is positive for amphiphilic secondary structure. So the hydrophobic moment is positive for amphiphilic secondary structure, and amphiphilic means they contain both hydrophobic and hydrophilic amino acids. Okay, and hydrophobic moment is, so here the hydrophobic moment is positive for amphiphilic secondary structures of protein. So let's move on to the next question, that is question number 85. So generally dash Packard sugar residue are formed in a DNA structure, and options are option A, C3 prime exo, or B option B, C3 endo, or option C, O4 prime exo, or option D, C4 prime endo. So the right answer is option B, that is C3 prime endo. So generally C3 prime endo Packard sugar residue are found in aDNA structures. Okay, so let's move on to the next question, that is which is an example of metalloprotein. So options are cytochrome, myoglobin, option C, hemoglobin, or option D, all of the above. So the right answer is option D, that is all of the above. So let's move on to the next question, that is question number 87. So here the bond present in lipoproteins and glycoproteins is covalent or hydrophobic or van der Waals or all of the above. And the right answer is option A, that is covalent. So the bond present in lipoproteins and glycoproteins is covalent bonds. Okay, so let's move on to the question number 88. So that is hydrolysis of ATP over ADP and AMP generates highest energy because ATP on hydrolysis generates thermodynamically stable structure, or option B, that is ATP is highly unstable, option C, hydrolysis of ATP is pH dependent, or option D, hydrolysis of ADP and AMP do not generate thermodynamically stable structure. So the right answer is option A, that is ATP on hydrolysis generates thermodynamically stable structures. So hydrolysis of ATP over ADP and AMP generates highest energy because ATP on hydrolysis generates thermodynamically stable structure. Okay, so let's move on to the next one. Which of the following reveals the 3D assembly of two structural units to form mature segment of polypeptide and its component domain? And options are primary structure, secondary structure, tertiary structure, or quaternary structure. And the right one is option C, that is tertiary structure. So tertiary structure. So the 3D assembly of two structural units to form mature segment of polypeptide and its component domain is tertiary structure. So tertiary structure is involved with 3D assembly of polypeptide chain. So let's move on to the question number 90. That is, which of the following reveals directly the folding of short segment of polypeptide chain into geometrically ordered unit? And options are primary structure, secondary structure, tertiary structure, or quaternary structure. And the right answer is option B, that is secondary structure. So secondary structure reveals directly the folding of short segment of polypeptide chain into geometrically ordered unit. Okay, so next question is question number 91. The primary difference between glycogen and cellulose is in the option A, type of constituent monosaccharides, or option B, number of monomers per molecule, or option C, configuration of the monomer, or option D, susceptibility to acid hydrolysis. So the right answer is option C, that is configuration of the monomer. So the primary difference between glycogen and cellulose is in the configuration of the monomers. Okay, so next question is, which one of the following repetitive motifs is responsible for the formation of triple helix in collagen? And options are option A, alanine XY, or B, glycine XY, or C, cysteine XY, or D, proline XY. And the right answer is option B, that is glycine XY. That means glycine and any two amino acids. Then these repeating units are always seen in collagen. So this glycine XY repetitive motifs is responsible for the formation of triple helix in collagen. Okay, so let's move on to the question number 93. So which of the following processes occurs in the formation of disulfide bridge between cysteine residues? And options are option A, reduction of sulfhydryl group, or option B, electrostatic interaction, option C, oxidation of sulfhydryl group, or option D, hydrogen bond formation. And the right one is option C, that is oxidation of sulfhydryl group. So oxidation of sulfhydryl group, which is a which which is present in disulfide bridge between cysteine residues. Okay, so let's move on to the next question. That is, active site of all serine protease consist of option A, serine, glutamic acid, aspartic acid, or serine, glutamic acid, methionine, or serine, histidine, aspartic acid, or alanine, glutamic acid, and methionine. And the right answer is option C, that is serine, histidine, and aspartic acid. So active site of all serine proteases consist of serine, histidine, and aspartic acid. Okay, so let's move on to the question number 95. So conversion of glucose to glucose 6 phosphate requires energy. However, critically ill patients are treated with intravenous infusion of glucose rather than glucose 6-phosphate? Because option A, glucose 6-phosphate is unable to enter into cells. Option B, glucose 6-phosphate is degraded very fast. Option C, exogenous glucose 6-phosphate is toxic to the cells. Or option D, exogenous glucose 6-phosphate will competitively inhibit endogenous enzyme. And the right answer is option A. That is, glucose 6-phosphate is unable to enter into cells, and that's why critically ill patients are treated with intravenous infusion of glucose rather than glucose 6-phosphate. Okay, so let's move on to the question number 96. So, which of the following reveals the number and type of polypeptide unit of oligomeric protein and their spatial arrangement? And the options are primary structure, secondary structure, tertiary structure, or quaternary structure. And the right answer is option D, that is quaternary structure. So quaternary structure reveals the number and type of polypeptide unit of oligomeric protein and their special arrangements are present. Okay, so next question is, which of the following is or are angles that define commonest secondary structure of protein? And options are option A, alpha helix, option B, beta sheet or beta helix, or option C, both A and B, option D, neither A nor B. And the right answer is option C, that is both A and B is correct. So alpha helix and beta sheet both are present in secondary structure of protein. Okay, so let's move on to the question number 98. That is, urea and guanidine hydrochloride are potent protein denaturants. So they work by binding to the hydrophobic groups in proteins, or breaking of disulfide bonds, or enhancing the hydrogen bonded network of water, or binding to the peptide bonds of protein. And the right answer is option D, that is binding to the peptide bonds of protein. So urea and guanidine hydrochloride are potent protein denaturants, and they work by binding to the peptide bonds of proteins. Okay, so next question is, an enzyme has two binding sites for an inhibitor molecule. So when the inhibitor binds to the first site, the dissociation constant of the inhibitor for the second site increases, so leading to the negative cooperativity. So the Hill coefficient for such an inhibitor is option A, equal to 1, option B, greater than 1, option C, less than 1, option D, less than 0. And here the Hill coefficient means the it is a measure of cooperativity binding of protein, okay, or enzyme. So the answer is option C, that is less than 1. So in this case the Hill coefficient for such an inhibitor is less than 1. So let's move on to the last question of this video, is, which of the following diseases are associated with genetic or nutritional deficiencies that alter protein maturation process? And options are mad cow disease, option B, Alzheimer disease, option C, bovine spongiform encephalopathy, or option D, all of the above. And the right answer is option D, all of the above diseases are associated with genetic or nutritional deficiencies that alter protein maturation process. Okay, so these are all 20 questions on biochemistry in this part 5 video. Okay, so thank you for watching this video.